but still average 600 is coming.
Every day.
So every day, sir, every day. And in the campus also situation is kind of very much stable. So yeah, so they are doing all kinds of testings, antibody, antigen, PCR, so all kinds of testings are going on.
What about the functioning of the institute? People are coming.
Yes, sir, that the most of the faculty members and the staffs are coming, but not the students, sir.
Students are not there.
Yes, sir.
So regularly it is going on.
Yes, sir. Yes, sir. Regularly, all the employees and the other essential service, everything is going on, but not the students. So maybe they will be coming, maybe more a little bit situation when it will be stable. So then they will be coming up probably. Because in the city, so there are some cases are there, and it is also growing up. So that's why for the student that restriction is there. Okay. But let's see how it is going. Yeah.
Okay, it's 2:30, sir. Shall we start?
Yes.
Right.
Good afternoon, sir. Good afternoon. Passenger, sir. Good afternoon.
Good afternoon. Good afternoon. Good afternoon to everybody.
So a very warm good afternoon to all of you. Today we are into the 8th day of our National E-Conference, and today we have our eminent guest speaker, Dr. Prasenjit Manna, with us, Principal Scientist of the Biotechnology Group and Biological Science and Technology Division, CSIR, Northeast Institute of Science and Technology, Jorhat, Assam. I welcome you, sir, to this event.
Thank you. Thank you very much, Dr. Finos.
Next, I'd like to welcome Dr. B.G. Unni, sir. He's our advisor as well as Research and Academics Director. I welcome you, sir.
Thank you.
Next, I'd like to welcome Dr. Naveen Mohan, who is our principal and also the convener of this event.
Thank you, sir. Thank you.
Next, I'd like to welcome all the participants from all over the country who have spared the time to participate in this event. Next, I'd like to welcome Ms. Radhika R. Krishna, Head of the Department of Microbiology, for introducing the speaker to this audience.
Good afternoon to all. It's my honor to introduce today's guest, Dr. Prasenjit Manna, Principal Scientist, Biotechnology Group, Biological Science and Technology Division, CSIR, NIST, Jorhat, Assam. He received a PhD in molecular medicine from Bose Institute, Jadavpur University, Kolkata, 2010. Dr. Manna has a successful track record comprising experience, commitment, and impact publications in understanding basic molecular mechanisms, conducting studies with diabetic animals and dogs, as well as environmental toxin-induced organ pathophysiology. Dr. Manna has published 64 articles in peer-reviewed journals and has an HS index of 26 above science as of May 2015. Dr. Manna has years of experience in research on streptozotocin-induced type 1 diabetic animal studies. Dr. Manna's work suggested the beneficial role of a bioactive natural product against streptozotocin-induced diabetes and its associated pathophysiology in rats. In addition, he has also investigated the mechanism of environmentally exposed nanostructure-induced organ pathophysiology. He has long experience in isolation and characterization of bioactive molecules from traditional Indian medicinal plants. During his postdoctoral fellowship in the lab of Dr. Kane, 2010, Dr. Manna has worked on the role of hydrogen sulfide and glutathione in mediating the effect of L-cysteine and vitamin G in insulin signaling pathways using monocyte, PTCL1 adipocyte, C2C12 myo2, and L6 myo2 cell culture models. Dr. Manna's recent studies have investigated the role of membrane-bound phospholipid in high glucose-induced impaired glucose metabolism, vascular inflammation, and endothelial cell dysfunction. Once again, I welcome Dr. Prasenjit Manna for the presentation. Thank you.
Thank you, Radhika Krishna. The floor is open for presentation.
Oh, okay. So, I'll be going to. It is showing Dr. Finoj is showing that the Dr. Finoj is presenting. So should I click?
Presentation. Yeah.
Okay, so I can go it now. Yeah.
Yes, sir.
Yeah. Yes. Okay. Now, go on. Yeah. It's okay.
Okay, sir.
Yeah.
Okay. Okay. Respected Dr. Uni and Principal Dr. Mohan, and also the organizing committee. At first, I would like to thank you very much for inviting me and in your excellent e-conference on current trends in life science, and for me to give a presentation in this institute. It will be like the very much honor. And it is always kind of very good to interact with the students in the undergrad college and also the master students also. And so today I'll be talking about some new perspective of an old vitamin. And so before going to my topic, I would like to discuss about the functioning of these kind of micronutrients in the management of disease mechanisms. So, and also before going to my presentation, I would like to acknowledge my director, Dr. J.N. Sastry, and also I'd like to acknowledge Dr. B.G. Uni, who is also every time which giving me so many supports and encouraging my research works and everything. And also I'd like to acknowledge Dr. Jyotin Kalita. He's also one of my senior colleagues, which supporting me, and my students, Mr. Onjum Dengia and Miss Jikasha Bardoli. And also my collaborators, Professor Paramesh Sheel and Professor Shaikat Dhyanji. And I would like to acknowledge DBT and DST for my funding. And we have declared that no conflict of interest exists in our study. So, micronutrients, they are playing an important role in our body for a specific actions. So, a very small quantities of the macronutrients, it is required to maintain the specific function, but the moderate deficiencies of them can lead to serious health issues. So initially in the nutrition community, they're focused on the role of macronutrients in preventing their deficiencies associated diseases. But with the time, it becomes clear that these nutrients can play an important role in a broader ways. So micronutrients like say vitamins, minerals, amino acids have been increasingly used in various healthcare deliveries due to their importance in the disease management. So, even initial depletion of them due to say inadequate sufficient supply, or maybe due to the increased absorption, it may lead to the alteration in biological function, which is associated with say oxidative damage, defects in metabolism, and also some non-specific functional defects like as immunological disorders, which may lead to clinical diseases. So vitamin K, it is a group of compounds. It is not a single compound. It is a group of compounds with the common structure of say 2-methyl-1,4-naphthoquinone, and it is an important like the vitamin K is known for its anti-hemorrhagic factors. So there are three forms of vitamin K. One is the vitamin K1, which is known as the phyloquinone, found in the plant foods. So like this is the example of say vitamin K1, and where you can see that the apart from its methyl group, it has some like the phytyl side chain, and with a like the trans double bond here. And so this is the vitamin K1, or commonly known as the phyloquinone. And next is the vitamin K2, which is the minaquinone, mostly found in the animal and the bacterial sources, where many more unsaturations are there. And third one is the synthetic form of the vitamin K, that is the vitamin K3, which is known as the minadione. But here there is no side chain like this, phytyl side chains are not there. So what is the recommended amount for this vitamin K? It is the for the newborn baby, it is the 500 to 1000 microgram once. So one thing I would like to mention that, so after the birth, say at a single dose of like vitamin K has been given because the vitamin K has not been transferred from the mom to baby by the placenta. So after birth, the vitamin K injection should have been given to the baby to prevent this blood coagulation. So for this adult, around 80 mg per kg body weight per day, this is the required dose. And for say 1 to 6 month kids like baby, so it is the 5 milligram per day. And for 6 to 12 months, it will be given 15 milligram per day. So what are the rich sources of vitamin K? This is spinach, green cabbage, parsley, lettuce, beef liver, green tea, and so this is the like the common structural unit of the vitamin K. And it has been found that the methyl group at the C2 position and the unsaturation of the phytyl side chain at the C3 position. So any alkyl group larger than the methyl present at C2 position will decrease the activity of the vitamin K. Simultaneously, hydroxylation at position C3 or any saturation or cis configuration of the phytyl side chain will reduce the activity of the vitamin K. However, if position 1 and 4 are substituted with say hydroxyl group or acetylation or say like the ether formation, so like this will not affect the activity of the vitamin K, that is the 1 and 4 position. So most important position are the C2 and C3. So vitamin K is well known for its critical function in activating the blood coagulation factors during the process of the homeostasis. So after the dietary intake, let's say, like from our dietary intake, we are getting this vitamin K quinone moiety. So in the presence of NADPH dependent quinone reductase, the like as I have mentioned that the double bond O at the C2, 1 and 4 position, so they have been reduced and this vitamin K hydroquinone has been formed. So this is the main ingredient for the functioning of vitamin K. So in the presence of the enzyme gamma glutamyl carboxylase, vitamin K is oxidized to the vitamin K oxide. But at the same time, like it will convert the vitamin K dependent protein, like this glutamic acid residues will be converted to the gamma glutamyl, like the gamma glutamyl carboxylic acid. Like in this, like the glutamic acid, so there, like the another carboxylation will be like will occur in this that the glutamic acid residues. So what will be happening? So like the presence of this 2 carboxylic group will help to chelate with the calcium and which will help for this blood coagulation process. So after that, vitamin K will be again reduced to this vitamin K quinone with the presence of this say vitamin K epoxide reductase. So this cycle is continuously going on inside this liver because the most of the vitamin K dependent proteins are present in the liver, and to maintain this like the level, this cycle is constantly going on. So like to prevent this blood coagulation, like the amount of vitamin K is required is very small amount, and this that's why this cycle is going on, and it is not any problem is happening unless and until the enzyme activities are kind of not like hampered by the access of any diseases. But what interesting thing is that most of us we are getting enough vitamin K from our diet to maintain the like the blood clotting, but it is not enough to offer protection against the various health problems such as cardiovascular diseases, osteoporosis, infectious diseases, like, and also this many new diseases are coming up. So in our recent studies, we have observed that the vitamin K supplementation, it is improving this insulin sensitivity, regulating the glycemic status, and as well as reducing the risk of type 2 diabetes. And the coagulation unrelated, so the proteins, whatever I have discussed, that is the vitamin K dependent GLA-rich protein, so the most of them are involved in this coagulation process. But there are some like the coagulation unrelated proteins such as GAP6, matrix GLA protein, growth at specific protein, so they are gaining scientific attention for their role in regulation of the metabolic syndrome as well as its associated disorders. However, there is no previous study which has examined this association of vitamin K as well as vitamin K dependent proteins with impaired lipid homeostasis and its associated inflammation. So what is hyperlipidemia? So hyperlipidemia, it is defined the elevated levels of circulatory lipids such as triglyceride and cholesterol more than 4 million, and also it accounts more than 4 million deaths annually in the world. And it is ranked as one of the greatest risk factors contributing to the prevalence and the severity of the coronary heart diseases and atherosclerotic cardiovascular diseases. Moreover, American Heart Association has identified these elevated blood levels of cholesterol and triglyceride are the primary risk factor for obesity and type 2 diabetes. In patients with hyperlipidemia, many signaling pathways involved in lipid metabolism are dysregulated and leading to these aberrant plasma lipids and lipoprotein profiles. So, in our study to examine whether this vitamin K supplementation has any effect in regulating this lipid metabolism or not, we have supplemented the vitamin K to the cell culture models, so like using the hepatocyte cells. Because the liver, it plays a central role in lipid synthesis and metabolism, so that's why we have chosen the hepatocyte cell culture model. This is the cultured hepatocytes. What we have done, so we have treated the cells with the vitamin K different concentrations, say 1 nanomolar, 5 nanomolar, 10 nanomolar, and for the 2 hours. After that, we have treated these cells with the palmitic acid, which is a model for the free fatty acid at a dose of 0.75 millimolar. This is a very high concentration of the palmitic acid. After that, we have scrapped these cells, and they are also used to check this cell viability assay, and also we have examined, say, triglyceride assay as well as intercellular lipid levels also. So what has been observed? So this is the cell viability data, and this is the intercellular triglyceride level. What you can see that the treatment of this vitamin K as well as the lipids, that is the palmitic acid, so none of the treatment caused any change in any significant changes in the cell viability. So it says that we are not disturbing the pathophysiology of the cells, so they all are fine. But interestingly, treatment of this palmitic acid, the free fatty acid, it causes an increase in the intracellular triglyceride level compared to the control. However, treatment with the different concentration of the vitamin K, such as 1, 5, and 10 nanomolar, it definitely decreased this intracellular triglyceride level. So next we examine this intercellular the lipid accumulation by the confocal microscopy, and we have stained these cells with the Nile red, which will bind with the neutral lipids, and also the nucleus has been stained with the DAPI. So in the fatty acid treated group, you can see there is an increased expression of this Nile red staining cells, that is the red colored cells, and treatment with this vitamin K against this palmitic acid, it gradually decreased this red color stain. So that we have plotted this like this counting Nile red versus DAPI, because the DAPI staining has been normalized, and then you can see that this treatment with the vitamin K, it gradually decreased the intracellular Nile red staining compared to the fatty acid treated group. So it also suggests that vitamin K supplementation dose dependently reduced the accumulation of the lipid droplets in palmitic acid treated hepatocytes. So to investigate what is the signaling mechanism playing a role in regulating this lipid metabolism pathway, so according to the literature survey, it has been mentioned that the free fatty acid, treatment the free fatty acid causes a decrease in the protein expression of the phospho-AMPK, that is the AMP activated kinase, and downregulation of the AMPK, it causes the upregulation of the SREBP1, that is the sterol regulatory binding protein 1 as well as the fatty acid synthase, and upregulation of both SRBP1 and FAS1, they increases the de novo lipogenesis. On the other hand, downregulation of the AMPK and causes the downregulation of the PAR alpha, which is a nuclear receptor, and regulates the fatty acid oxidation pathways. And so downregulation of this PPAR alpha causes the downregulation of the CPT1A, which is nothing but the genes involved in the transfer of the fatty acid to the from inside the mitochondria, and it also causes the downregulation of the UCP2, that is the uncoupling protein 2, also involved in the fatty acid oxidation process. So fatty acid exposure to the cell, so they causes the downregulation of this AMPK, and which is leading to the upregulation of this de novo lipogenesis and regulation of the fatty acid oxidation. So in our study, we have found that vitamin K treatment against this palmitic acid exposure, it causes the upregulation of the phospho-AMPK, upregulation of the phospho-AMPK compared to the palmitic acid treated group. Parallelly, it downregulates the SRBP expression, SRBP1 expression, as well as the downregulate the FAS protein expression. You can see this band, that is the treatment with this vitamin K against this palmitic acid exposure. It causes the downregulation of the protein expression of FAS, which is nothing but the fatty acid synthase protein. So the lipid exposure, which is causing the downregulation of the AMPK, as well as this upregulation of this SRBP1 and FAS1. So treatment with this vitamin K, it causes the upregulation of the AMPK and downregulate the SRBP1 and fatty acid synthase 1. And finally, it regulates this de novo lipogenesis. On the other hand, vitamin K supplementation, it also upregulates the PPAR alpha protein expression, as well as upregulate the CPT1A protein expression, that is the proteins responsible for the beta oxidation. So whatever the fatty acid is causing the downregulation of the beta oxidation of fatty acids, vitamin K supplementation, it improves this fatty acid oxidation pathways. So after that, whatever the results obtained in the cell culture treatment, we want to validate this hypothesis in the animal study. So what we have done, so like we have taken this albino mice, male albino mice, acclimatized at the animal house for 1 week, and we have classified the animals into 4 groups. So in the first 2 groups, one is the control as well as the vitamin K treated group, and in the another 2 groups, we exposed them with this high-fat diet. So high-fat diet, it contains 20% of like the protein, 35% carbohydrate, and 45% fat. So, and here olive oil is our vehicle, because we cover this vitamin K using olive oil as a vehicle. So we have treated the animals for 16 weeks against this high-fat exposure, and in the last 8 weeks, we have covered them with the vitamin K. After 16 weeks, the animals has been sacrificed, blood samples and liver tissues were collected for further experiment. So this is the serum parameters. What we have found that none of the treatment are causing there is significant increase in this ALT, AST level, that is the alkaline phosphatase aspartate transaminase, the indicator for this liver dysfunction. And also there is no changes in the significant changes in the creatinine level. So that means that the liver and kidney, they are kind of fine. But what is happening? So high fat diet exposure, it causes a significant increase in the triglyceride level as well as total cholesterol level compared to the control and groups. However, supplementation with the vitamin K, it reduces the triglyceride as well as the cholesterol level compared to the high fat diet fat group. And it suggests that the vitamin K supplementation, it can reduce your cholesterol level compared to your high fat diet group. Then we examined the histologically the liver tissues, and so what has been found that in the high fat diet group there is a significant accumulation of the lipid droplets in the liver tissue compared to the control, but that tissue has not been damaged, because there is not a significant increase in this ALT or leakage in the ALT or ALT enzymes, which suggests that this early exposure of this fatty acid, it causes this lipid accumulation, but this liver has not been damaged yet. So, and in this case, like if we supplemented the animals with the vitamin K, it reduced this intracellular lipid accumulation. So before it's too late, if you have some proper control in your hand, like say vitamin K, so which can prevent this lipid accumulation in the liver. So here this green arrows shows this lipid droplet appears in the liver tissues. However, this lipid droplets are very much less in this vitamin K treated high fat diet mice. After that, we have examined the same signaling molecule, whatever we have discussed in the cell culture studies. So, like say phospho-AMPK, you can see that in the high fat diet group compared to the control group, this phospho-AMPK level is very much less, almost absent. Treatment with the vitamin K against this high fat diet exposure, it increases this phospho-AMPK level. Similarly, treatment with the high fat diet, it causes the activation of the SRBP1 protein expression, as well as it also causes the increase in the mRNA expression of this SRBP1, which is the transcription factors and which regulates this de novo lipogenesis. So vitamin K supplementation, it reduced this protein expression of the SRBP1 as well as the mRNA expression. Similarly, the protein expression of say FAS, that is the fatty acid synthase enzyme, has also been increased in the high fat diet fat group, and also the mRNA expression of the FAS has been increased in the high fat diet group. Treatment with the vitamin K against this high fat diet, it reduced this FAS protein expression as well as the mRNA expression. Same study has also been reflected in the fatty acid oxidation pathway also. So the high fat diet exposure, it reduces the PPAR alpha protein expression as well as the CPT1A protein expression and UCP2 protein expression, and the mRNA expression has also been reduced in presence of the high fat diet. However, treatment with the vitamin K against this high fat diet, it upregulates both the protein as well as the mRNA expression of the PPAR alpha, CPT1A as well as the UCP2. And we have also examined the activity of this fatty acid synthase enzyme as well as this beta oxidation activity. So high fat diet exposure, it increases the activity of enzyme activity of fatty acid synthase, as well as this decreased this the enzyme activities of CPT1A. However, treatment with the vitamin K, it decreased this enzyme activity of FAS and as well as increased this enzyme activity of CPT1A. So until this, it has been observed that this vitamin K supplementation, it is helpful in reducing these lipid levels in the blood as well as in the liver tissues. Next, we examined the vitamin K levels as well as these lipid parameters in the healthy control as well as the subjects with the hyperlipidemia. So what is our aim? We want to examine whether there is any correlation exists between the vitamin K levels as well as the lipid parameters, because so far we have understood that the vitamin K supplementation is helpful in reducing the lipid level. So we can expect that there may be a inverse relationship between the vitamin K level as well as the lipid level. That is increasing the vitamin K concentration in your blood may be helpful in reducing your lipid levels. So see what is happening. So we have taken say healthy control as well as the hyperlipidemic subject, and their body weight are kind of within the same range, and so the hyperlipidemic subjects, they have a significantly high levels of this triglyceride as well as the total cholesterol. However, their vitamin K level is lower than those observed in the healthy control. But whenever we want to examine the correlation between the triglyceride level as well as the vitamin K, what we have found that although an inverse correlation exists between the triglyceride as well as the vitamin K level, but it is not significant. So then it came to our mind that maybe our subject number may be less. That is because we have taken only the 22 number of subjects. So it may be the reason, or maybe it may not be like that, because it may not be the vitamin K which is playing a role in maintaining in regulating this triglyceride and total cholesterol level as well as the lipid metabolism in our body. Then we go back to our main hypothesis, that main function of the vitamin K, where it has been mentioned that the vitamin K, apart from the activation of the coagulation related protein, there are some different kind of coagulation unrelated protein, like say osteocalcin, matrix protein, growth specific protein 6, as well as the cartilage matrix associated proteins. It also activates those proteins, because those protein has also these glutamic acid residues. So we want to examine the levels of those proteins in the human subjects. So we have examined the levels of this GAS6, that is the growth addressed specific protein 6, because in the literature it has been observed that this GAS6 protein, it is highly related with your obesity as well as the lipid metabolism pathways. So we want to examine whether this protein has any role in regulating this lipid metabolism. And we found that, interestingly, there is between this healthy control and the hyperlipidemic subject, there is no significant difference between in the GAS6 protein level. However, when we examine this carboxylated gas 6 level, that is the vitamin K dependent activation of this gas 6, then we have observed that there is a significant difference between these C gases, that is the carboxylated gases level, between your healthy and hyperlipidemic subjects. So, in the same subject population, we did the correlation studies again. So now it has been, interestingly, it has been now observed that with the increase in the concentration of the vitamin K, it causes the increase in the concentration of the carboxylated gases, because this gas 6 protein, it has this glutamic acid residues which is getting carboxylated and forming the C gas 6, that is the GLA gas 6, in presence of vitamin K. So now like if we are performing these correlation studies between these carboxylated gases versus total cholesterol, we are now we observe that there is a significant negative correlation exists between these carboxylated gases and total cholesterol as well as the triglyceride. It is the same population where earlier we have examined the correlation between the vitamin K and total cholesterol as well as the triglyceride. We didn't find any significant relationship, although it was the negatively correlated, and at that point we thought that it is the vitamin K which is directly involved in this in regulating this cholesterol as well as the triglyceride level. But now we have observed that it is not the vitamin K directly, but it is the vitamin K dependent protein, that is the carboxylated gas 6, which is negatively correlated with your triglyceride as well as the total cholesterol level. So then we have we thought that how to examine this relationship in our animal as well as the cell culture model. So then we go back to our study again. So then we, in the same animal group, we examined this the GLA gas 6 as well as the total gas 6 level. So in the animal study, what we have found that in the total gas level in the high fat diet group and vitamin K treated group, there is no difference in this total gas 6 level. However, in the carboxylated gases, that is the GLA gas 6, vitamin K supplementation, it causes the upregulation of this GLA gas 6 level in the plasma. Now, we have examined the protein expression level in the liver tissues also, and by doing these immunoprecipitation studies, we have observed that supplementation with this vitamin K causes this upregulation of this gas 6 protein in the high-fat diet plus vitamin K treated group. However, total gas 6 protein expression, there is no change in the total gas 6 protein expression. So, after that, we have conducted this cell culture experiment again, and to examine this triglyceride level, nile rate staining, as well as the cell viability. Whatever the study we have done earlier with the vitamin K alone, now we are performing the same study with these carboxylated gases as well as this uncarboxylated gas 6, that is this recombinant gas 6. And we have found that they say, you can see that they say, whenever you are treating these cells with this fatty acid, that is the palmitic acid, there is a significant increase in the triglyceride level as well as this nile rate DAPI staining. And however, if you are treating the cells with this non-carboxylated gases, that is the R gas 6, there is not a significant change in the triglyceride level as well as the nile rate staining. However, if you are treating the cells with the either vitamin K or carboxylated gas 6 against this palmitic acid exposure, both of them are causing the significant downregulation of the triglyceride level as well as the nile rate staining. So, both these carboxylated gas 6 as well as the vitamin K, they are showing the same effect. That is the reduction in the triglyceride levels as well as this reduction in this nile rate staining. However, none of the treatment they are causing any changes in the cell viability. So again, we have examined this same protein expression level. So that is this phospho-MPK, SRBP1. So treatment with this both this vitamin K as well as the carboxylic acids, they are causing this upregulation of the phospho-MPK and downregulation of the SRBP1 protein expression, its mRNA expression, and also both this vitamin K as well as the carboxylic acids, they downregulated the protein expression of the fatty acid synthase as well as the mRNA expression of this fatty acid synthase protein. Okay, so like the it has been observed that the C gases, that is the carboxylic acids, and as well as the vitamin K, both of them has the similar kind of effect. And also the beta oxidation, that is the fatty acid oxidation pathway, we have observed the similar story, that is the vitamin K as well as the carboxylic acids. It causes the upregulation of this PPR alpha protein expression as well as its mRNA expression. Also this CPT1A protein expression has also been increased, its mRNA expression has also been increased, and the uncoupling protein 2's protein expression and mRNA expression both has been increased in presence of the vitamin K or carboxylic acids. However, in none of the treatment, that is this R gases, that is the recombinant gases, which is the non-carboxylated form, they didn't cause any changes in the protein as well as the mRNA expression of none of these signaling molecule compared to the fatty acid treated groups. So now, like if you can remember the say enzyme, so that is the from this vitamin K, that is the hydroquinone to the vitamin K epoxide. So there is an important enzyme, that is the gamma glutamyl carboxylase, that is the GGCase, which is like the presence of which the vitamin K is carboxylating this glutamic acid residues to gamma glutamyl carboxylate. So, like if we are knocking down that enzymes, and now we want to examine the effect of vitamin K as well as the carboxylic acids. So, interestingly, you see, when in the knockdown cells, that is the knocking down this enzyme, that is the GGCase, in the knockdown cells, vitamin K cannot activate this phospho-MPK, as well as it cannot downregulate this SRBP1, FAS1, that is this that is de novo lipogenesis, as well as it cannot activate this PPR alpha, CPT1A, as well as UCP2. So in the GGCX, that is the knockdown cells, vitamin K cannot activate these proteins involved in the de novo lipogenesis as well as the fatty acid oxidation. However, in these cells, in the knockdown cells, you treated with this that the gamma glutamate, that the carboxylated acids, it significantly upregulated phospho-MPK, it significantly downregulated SRBP1, as well as upregulated the PPR alpha, CPT1A, as well as the UCP2. So what it suggests? It suggests an important message, that is, the vitamin K, whatever the effect it is showing, it is mediated via its carboxylated proteins, and Cas6 is one of the important proteins which is involved in this process. So, in summary, we can say that the circulating levels of the vitamin K and carboxylic acids are significantly lower among subjects with the hyperlipidemic patients, and so carboxylated acids, like when you are applying the high-fat diet or free fatty acid, it causes the downregulation of the carboxylated acids, which is causing the downregulation of the phospho-MPK, upregulation of the SRBP1, fatty acid 1, increasing the de novo lipogenesis, and also downregulate this fatty acid oxidation pathway. However, vitamin K, application of the vitamin K, it prevents this pathway, and it is functioning by the activation of this enzyme, that is the gamma glutamyl carboxylate enzymes. So in summary, we can say that the vitamin K, it maintains the lipid homeostasis by activating this GAS6 protein and regulating this AMPK mediated SRBP1 FAS as well as this PPR alpha, CPT1A, UCP1 signaling cascade of hepatic lipid metabolism. So thank you for your attention, and I would like to, I'm happy to answer all of your questions if any. Thank you very much.
Thank you, sir. It was a really informative session, and the floor is now open for questions. I'd like to welcome Ms. Resna, Assistant Professor in Microbiology, to lead the session.
Good afternoon to all. Am I audible?
Yeah.
Yes, yes.
Thank you, sir. Thank you, sir, for your nice presentation. And some queries are there in our chat box. One of them is that whether heparin is the only one antagonist of vitamin K.
So like heparin, apart from the heparin, there is another one is that is the coumadin is that, which is the coumarin moiety, which is also functioning as an anticoagulant.
Okay. Okay, sir. Another one is, when will you give vitamin K for a patient?
Mm-hmm. So like vitamin K, yes, go ahead please.
When will you give the vitamin K for a patient?
So like thing is that like if they do have any problem in this like the coagulation unrelated coagulation, if they have any coagulation related complication, then you will not be giving the vitamin K. So recommending the vitamin, before recommending the vitamin K, you have to check their like the prothrombin time, as well as the, like say like the APTT, PT time, like all these coagulation factors. So if they do not have any problem associated with the coagulation, and also you have to check that whether these liver marker enzymes are fine or not. And after that, you can supply the, you can give this vitamin K, because in the very beginning I have mentioned that the like most of us, we do not have this coagulation associated problem, most of us, but these micronutrients like is the things are coming, they do have the effect beyond their like the classical function. Okay, so you need more amount to regulate this unclassical function. So like, okay, if you can, you may notice that like this a few days before, while this Horlicks, like women Horlicks advertisements are coming, so there that this vitamin K is giving, that the vitamin K image is coming. And apart from that, nowadays like okay, in every store, if you can find, so with the along with the vitamin D, vitamin K are combining with your calcium. Okay.
Okay, sir.
Yeah.
Then last question is that this type of vitamin K additional supplementation, they may cause any interaction with drugs of a person if he take any medicine?
Yeah, so like the so far no report has been there. So interaction with the vitamin K with any other medication, but as I mentioned, like if any medicine has any chances of this coagulation associated problem, so like you have to consider only those issues. Otherwise, so far no nothing, no any toxic effect has been seen. And interestingly, this like the therapeutic windows of vitamin K is very long, very long. So like up to like say 1000 mg, up to 1000 mg, like the doses has been tested. So no toxicity has reported, but still because we are consuming within this microgram level. which are consuming the microgram level. So if you are consuming, say like in a every day in an alternative day, so you will be avoiding so many complications. And most of the complications related with your say like the bone-related dysfunction, tooth decay, and your say atherosclerosis, and finally this diabetes. So like the K has a tremendous role. Okay.
Okay, sir. Okay. That's all about the queries.
Yeah.
I think the questions are over.
Yes.
Yeah.
Any more questions are there?
No, sir.
Any more doubts or any queries?
No, sir.
No, sir. No.
No.
Okay.
No, it's not there. Okay.
No, sir.
Let me speak about the brief talk. In fact, Prasenjit Manna gave an unknown, the known vitamins, old vitamin. So everybody thought that it may be something about talking about maybe something different. But it was something a very interesting topic. And if you would have talked about, directly the talk is given like vitamin K and, diabetes type 2, then it may be something different. So he made the secret. He kept the secret and explained it very well. As on today, type 2 diabetes, it's becoming a very common. Then in type 1, that one is on insulin dependent, this is not. But Prasenjit Manna, what I know, that he has got a significant contributions in this area that you must have seen from his explanation, the research work, what he has carried out. The relation between the vitamin K to other metabolic disorders and other metabolism and other, at the molecular level, what he has studied and correlated and finally derived a conclusion. That is something, an ideal example of how one has to go to the research. That means till you get a conclusive evidence and keeping a factor in mind, that's the most important. So he gave a very thorough example to the new, the young generation people who is entering to the research. How a simple topic when you take, how you can go through that and how you can solve your problem or the objective, what you are keeping in your mind before starting the work. So it was a very well crystal cleared, the experiments what he carried out with the conclusive, and I'm thankful to Manna. In fact, he is very much associated with a number of activities at RRL. I know I had a tough time to get him for about 1 hour because there are so many official formalities one has to follow. He's also taking care of the COVID laboratory there at the institute. So many responsibilities he has got, but still he took his time and very well explained, and it's a very informative talk to each and every faculties and as well as the student. Thank you, Prasenjit Manna, for spending your very valuable time and giving a very excellent talk. Thank you very much.
Thank you, sir.
Thank you, sir. Sir, next I'd like to just a minute, sir.
Sorry. Actually, I would like to mention at this platform, whenever I'm talking about the vitamin K, because in my postdoc, so now I have started the work on this vitamin D, and starting the work on the vitamin K in India. So far I don't know any people who are working on the vitamin K in India in these metabolic disorder associated problems. But the thing is that now time is coming. So if you it is my general suggestion to all of you, whenever you will be taking any vitamin D supplementation along with your calcium, so make sure that the vitamin K is present, because now those tablets are in the market. So do not consume vitamin D alone, because I will explain you, because whenever you are taking the vitamin D for your calcium absorption, Okay, so to make your bone more strong and functioning very well. But vitamin D will help you to absorb the calcium only. Unless and until you take this vitamin K along with the vitamin D, calcium absorption is nothing. So like taking the extra vitamin D along with the calcium, you are inviting your cardiovascular complication. Make sure that it is very, very clear. It is very clear from the clinician point of view. So like whenever you will be taking any vitamin D along with the calcium, make sure that you have your vitamin K along with that. So that's the thing I just interrupted to give this like the very clear message, so that it should be, because I think it is my responsibility to inform our people from this platform. Thank you very much.
Thank you, sir. Actually, it was a great piece of advice to all of us in the molecular level. You are the one to speak on the molecular level, sir. And thank you. And next, the floor is open for a vote of thanks. I would like to welcome Ms. Rasna NK to carry the session.
Okay, thank you, sir. Honorable dignitaries and distinguished guests and dear participants, on behalf of the Microbiology and Biotechnology Department of GEMS College, I feel immense pleasure to take this opportunity to propose a vote of thanks to our eminent speaker, Dr. Prasenjit Manna, Principal Scientist, Biotechnology and who spare the time, his valuable time to participate to this event with his informative talk. And I take this opportunity to specially express my deep gratitude and thanks to our academic director, Dr. B.G. Unni, and our principal, Dr. Naveen Mohan, and all other faculties of our college and the organizing committee of this webinar. And my deep sense of appreciation and thanks to all the participants who choose this to be live in this occasion and attended such session and made it a successful event. Thank you all. Thank you.
Thank you. Thank you very much, Dr. Reshma.
Thank you, Manna. Thank you very much.
Thank you, sir. Thank you. Thank you, everybody. Thank you, everybody. Thank you.
Thank you, sir. Thank you, Unni, sir. Thank you, Naveen, sir.
Yeah, most welcome.
Thank you, Manu, sir.